Welcome back, everyone. So we are going into part 4 of our topic 4, genetic blueprint and energy production. So we've already talked about ATP and all the chemical reactions in our body and things like that. So we're going to start getting into exactly how we take our glucose, that nutrient that we obtain from, breaking down our carbohydrates, and extracting cellular energy from that to get that ATP. So we're going to start out by learning about the process of glycolysis. So by the end of this video, you should be able to describe the overall result in terms of molecules produced by the breakdown of glucose by glycolysis. So basically, you should know the inputs of glycolysis and the outputs of glycolysis. So before we get into the whole inputs, outputs, all of that fun stuff, I just want to give you an overview of what we're going to be talking about in the next couple of videos. Okay, so we're going to be getting into, again, how cells obtain their energy. So all cells require energy, right? And so they obtain it from the nutrients that they absorb. And our plants and animals, they undergo a process known as cellular respiration. Now this process occurs partly in the cytoplasm, but also with the help of the mitochondrium, that organelle that we talked about way back in topic 2, which is that powerhouse of the cell. Okay, so for this first part, we're going to talk about glycolysis, which is one of the steps of cellular respiration. And this first step occurs in the cytoplasm, or what we call the cytosol of the cell. So this actually takes place outside of the mitochondrium. And all cells do this, okay? It's not just our human cells or just plant cells, it's all cells. So even bacterial cells do some form of glycolysis. But just before we get into everything, okay, cellular respiration, which we're going to talk about with plants and animals, requires glucose, okay? So this is going to be one of our inputs, as well as oxygen. And then we'll talk a little bit about what ADP and PI is. So ADP is that adenosine diphosphate, which we talked about earlier, and then the extra phosphate. And then the outputs at the end of the reaction, we breathe out that carbon dioxide and water, but then we end up with 36 ATP at the end, okay? About. You'll see it range in numbers depending on where you look. Like this one, it says if you add them up, 2, 4, that's only 29. So it just kind of depends on which book you're using and so forth. But for this particular video, we're just focusing on glycolysis. So this is just the first step of the process, okay? So again, we are going to be learning about glycolysis, and this is going to be the first step of cellular respiration that's carried out by eukaryotes. Not, some organisms will just do glycolysis. And so that's why both prokaryotes and eukaryotes carry out some form of glycolysis. And glucose, we know glucose already is that, if we go back a slide, is that chemical formula C6H12O6. So when we break the bonds that hold this molecule together, we release energy. Okay, so we're going to harness that energy from breaking these bonds and use that to generate the ATP. Okay, so by breaking those bonds, we obtain the energy. Okay, so since all living things, from prokaryotes to eukaryotes, utilize glycolysis in some form or another, we think that, it must have evolved very early in life's history. Okay, so we talked about life's history early in topic 1. So, way back then when we just had, the primordial soup of life, we think that glycolysis evolved very, very early on. Okay. And again, that's just the first step. So basically we're going to take that glucose molecule that we've broken down from our food, and we're going to break it down and extract some energy from it. Okay. And again, like I said before, this takes place in the cytoplasm. So that's why it's present in all cells, because not all cells have mitochondria, like the prokaryotes. Okay, so this takes place in the cytoplasm of the cell. Now, before we get into glycolysis, and actually your book doesn't talk about this for some reason, you need to know about it, but your book seems to have skipped this important part. So I'm going to bring it up. Electron carriers. Now, throughout the process of glycolysis and the other steps of the of cellular respiration, you're going to see the terms NADH and FADH2. So we call these electron carriers. So what are electron carriers? So electron carriers, they're exactly what they sound like. They carry electrons. So basically they shuttle electrons by binding and carrying these high energy electrons between compounds in biochemical pathways. Okay. So when we have electrons that are added to a compound, we call it reduced. Okay, so when electrons are added to a compound, it's a reduction type reaction, so it's reduced. When electrons are stripped from that compound, it's oxidized. And a way to remember it, there's actually two acronyms that you can use. One of them I like better, so I'll use that one. It's called oil rig. So oil rig, so oil, think oxidation is losing electrons. So O stands for oxidation or oxidized, and then I is L, losing. So oil, oxidation is losing. And then rig, reduction or reduced, I is G, gaining electrons. Okay, so oil rig is an easy way for you to remember which is which. Okay, so when when a compound is reduced, it has gained the electron, so it's the rig part. When a compound has lost its electron, it's the oil part, so it's oxidized. Okay, so you'll see throughout the next two videos, we're going to see how we have an oxidizing agent, so something like NADH, or NAD+, excuse me, that will be around to gain electrons. So when it gains electrons, what is it called when it gains electrons? It becomes reduced. And when it's reduced, it turns into NADH. Okay, so it's gained an electron to form a compound or a molecule called NADH. Okay, same with another type of molecule called FAD. FAD is an oxidizing agent. When it gains electrons or is reduced to FADH2, that means that it's carrying electrons. Okay. And then once it is in this form, it's carrying electrons, right? So it has extra electrons, and eventually we'll see later on in the next video, it will drop off those electrons, so it will lose electrons. It will be oxidized. When it loses electrons, it'll go back to its oxidized form and turn back into NAD+. Okay, so I want to bring up these electron carriers just because you will see them in the throughout this video and in the next video, and actually the video after that. So NADH and FADH2 are considered electron carriers. In this form, they're carrying electrons. Okay, when they're NAD+ or FAD, they are not carrying electrons. Okay. So just be aware of those. And these, by the way, they have fancy names, but you don't need to know the names. It's like nicotinamide adenine dinucleotide is NADH. And gosh, what was, I can't remember, but so yeah, it's quite a long name for each one of these. So that's why it's abbreviated. So you don't need to know like the actual name, the full name, just the abbreviation is appropriate. Okay, so let's talk about actual glycolysis. So when we're talking about glycolysis, we actually have two phases of glycolysis. The first phase is what we call the energy investment phase. So we have to invest some energy actually. And then we have the second phase is the energy payout phase, when we actually get some energy back. We pay it out. So let's take a closer look at them. Now remember, the input of cellular respiration is glucose. So here's our glucose. And remember, glucose is C6H12O6. So there's 6 carbons, right? So here's our 6 carbons. So when we go into glycolysis, that energy investment phase means that we need a little bit of energy, some ATP, to break apart the glucose. Okay, so we need some energy to break these bonds. And so that's why 2 ATP are used. So you can see here, we have 2 ATP are going to be used. And when 2 ATP are used, that energy that comes from that reaction, okay, ATP is transformed to ADP. And remember that video we saw from the last, that Amoeba Sisters video from the last video, we saw how when that phosphate gets broken off, a burst of energy is released. So that's what helps break this bond here. And then we have ADP is formed, okay, with an extra phosphate kind of floating around somewhere else. Okay. So we have 2 ATP is used to break the bonds. Okay, so that's the energy investment phase. Okay, now we end up with the energy payout phase. So we end up with, we get these 3 carbons with that phosphate attached, and then we end up getting the payout. So the payout ends up with, we get these 3 carbon molecules, which we call pyruvate. So since we started with a 6 carbon ring, we end up with 2 3 carbon molecules, or 2 pyruvates. Okay, so we end up with 2 3 carbon pyruvate molecules being formed. So that's one of the outputs. Okay. In the process, we also see that NADH form. Okay, so we do see that NAD plus gets reduced. Remember, reduced RIG, reduction is gaining electrons. So during this process where we have the 3 carbon plus the phosphate undergoes the reaction, we end up gaining some electrons, and that electrons get added to the electron carrier NADH. So we have that happen once and twice, okay, because we have 2 pyruvates. So we end up with 2 NADH, or electron carriers. We also end up with 2 ATP for each pyruvate, okay? So since we end up with 2 ATP, that's 4 ATP total, okay? But because we use 2 ATP at the beginning, we have a net gain of 2 ATP. So we ended up with 4 ATP, but we used 2 ATP at the beginning, so 4 minus 2 was a net gain of 2 ATP. Okay. So again, we started out with glucose. We used 2 ATP. We ended up with 2 pyruvate molecules and 4 ATP and 2 electron carrier NADH molecules. So here is a summary of the process. By the way, I will include these summaries with all of our videos just to give you kind of a view of the inputs and outputs of each of the processes. And this might help you, especially when that part of the assignment in your workbook where you do that little game with the currency. So that might be helpful with you to kind of keep track of what is going into each of these reactions or steps and what is coming out. So in our inputs, we started out with that glucose molecule, that 6-carbon molecule. We also needed 2 ATP, okay? If you go back a slide, we did have 4 ADP hanging around, okay? And then we had 2 NAD pluses, okay? So those were the oxidized form. At the end of glycolysis, that 6-carbon chain got split into 2 3-carbon molecules, which we call pyruvate. So we have 2 pyruvate molecules. We have that 2 ATP became 2 ADP, okay? That 4 ADP became 4 ATP. So if we add it all up, we gained 2 ATP total from this whole process. And then the 2 NAD plus got 2 electrons to form 2 NADH. All right. So that is the inputs and outputs of glycolysis. So let's do a review question, make sure we understand it. The glucose that enters the glycolysis pathway is split into 2 molecules of, is it A, pyruvate, B, ATP, C, phosphate, or D, NADH? If you chose pyruvate, I would agree with you. So remember glucose, 6-carbon, and we split it into 2 3-carbon pyruvate molecules. All right, so by now you should be able to describe the overall result in terms of molecules produced of the breakdown of glucose by glycolysis. So that's just the first step of cellular respiration. So in the next video, we're going to learn about the Krebs cycle and electron transport chain.